So in Unit 3.2, we're talking about protein synthesis, and we're starting on page 49. Protein synthesis. A couple things we need to remember there. A couple little notes that I want you to add to the side. Remember that synthesis means to make or build. It was one of our Sturger life processes, one of the 8 functions that all living things need to perform in order to be considered living. Now, protein synthesis, making or building proteins, actually will occur in 2 steps. So today we'll actually focus only on the first step, which is called transcription, and tomorrow we'll focus on translation, the second step. So today our main goal is actually to talk about how do we get the information from the nucleus of the cell to the organelle that builds proteins, which is called the ribosome. So why is protein synthesis so important? Every inherited trait is controlled by one or more proteins. And a trait is any characteristic you could have received or inherited from your parents. For example, your blood type. There could be some sort of genetic disease. There could be your height, your eye color, your hair color, your skin color. Almost anything and everything that makes you who you are is usually inherited, and it's controlled by proteins. So each cell must actually produce a different protein based on the function of that cell. What is the cell's job? What proteins does it need to perform that job? So you can also think of this as the needs of the cell. So please include this. The needs of the cell will determine which proteins need to be made. An example is that your red blood cells need to produce the protein hemoglobin. Here's a picture of the hemoglobin in 2 different forms. Okay? Hemoglobin is the protein that helps to build or helps to carry oxygen in your red blood cells. So within your red blood cell, you have this protein. This protein allows you to carry oxygen throughout your body. So carrier of oxygen throughout the body. Without that protein, your red blood cells cannot carry oxygen. Then you can perform functions like cell respiration, aerobic cellular respiration, where you need oxygen to be able to produce ATP. So DNA is going to control the production of the proteins. Remember that DNA controls every process in your body. That's where all the information is stored. A small section of DNA, a small piece of DNA, is called a gene. And it is used as the blueprint for making a protein. So for example, in a picture of a chromosome, you have the chromosome being unwound. Remember that a chromosome is just DNA that's condensed or wound up in a neat package. So when we unwind it, we can actually see a small section of DNA with the A's, the C's, the G's, the T's. That's actually going to be a gene. And a gene is going to code for a very specific protein, a very specific trait. So let's add that in there as well, for making a protein or traits, because your traits are controlled by your proteins. Each gene is composed of a specific sequence of nucleotides. For example, the DNA sequence for insulin would be CCA, TAG, CAC, etc. Okay? And it can be a really, really long sequence of DNA or a shorter sequence of DNA, just depending on what information you need or how much information you need. The cell knows what protein to make based on the sequence that it receives from the DNA. Therefore, the order of the bases matter. Very important here. If the order of this DNA sequence for insulin was changed, possibly by just 1 letter, maybe 1 letter was missing or an extra letter was put in, you can actually make a completely different molecule, but you'll definitely not be making insulin properly. If you don't make insulin properly, then that can lead to diseases like diabetes. So here's the central dogma of biology. And that just means the main idea, this main theory that we believe in, what we hold as true. And so the central dogma of biology actually describes the sequence in which information is passed to express your traits. And again, your traits are controlled by proteins. This picture you see on the board is a little different from what you see in your notes, but I want you to try to fill in these boxes, and we'll double check tomorrow in class. But in your first picture, you have DNA, and notice that there's this little kind of circling structure on the DNA, part of the diagram, kind of circles around like this. Well, if you take DNA and you make more DNA, remember that this is called what? It starts with an R. This is actually going to be replication. Replication. Okay? So make sure you're looking at your picture and my picture and comparing. But after DNA is replicated, and sometimes it'll be replicated before we do protein synthesis, and sometimes it won't be, but you have DNA, and now DNA is going to go through a process called transcription. And transcription is where you make some mRNA. On your notes, you only see the RNA, and I want you to underline the mRNA, or add the M in. Okay? So transcription is this first step to protein synthesis. Then when you have your mRNA, and again I want you to add in the M, you'll go through a second process, or second step, to actually produce the protein. And this step is called translation. So today what we're actually going to do is we're going to focus on transcription. What's important about this central dogma? Well, the central dogma is basically saying, how do you pass on information? The information starts with your DNA, goes to mRNA, and from mRNA, we can have that protein being produced. Okay, so make sure you fill in those three boxes. Sequence of information and how it is passed. So in protein synthesis, we actually will require RNA, and this is actually section D in your notes. There's a little typo right here. D. There are 3 types of RNA that we'll need to know for this process. We have mRNA, which is messenger RNA, tRNA, which is transfer, and rRNA, which is ribosomal, actually makes up the ribosome. So RNA is actually made up of a single strand of nucleotides, and the nucleotides consist of the ribose, phosphate, and nitrogen base. Basically, RNA is made up of the same type of nucleotide as DNA. And one thing to keep in mind before we move on is that RNA, the 3 different types of RNA, actually have 3 different shapes or structures. And this is an example of what tRNA looks like. mRNA, or RNA itself, is single-stranded nucleic acid. And what we're showing you in this picture at the top is that we actually write down the sequence of those nucleotides in sets of 3s. And it's really important. Notice before, in your notes for the sequence for insulin, we also broke up those letters in sets of 3s, or triplets. So in RNA, uracil places thymine. Thus, A pairs with U, and G pairs with C. So if you had a big, big, long sequence of it with AAAs, side to it, like DNA when it pairs up, will actually have U's instead of T's. That's all that's saying. And we'll talk about that more in class. Again, notice in the picture of mRNA that you have these U's and no T's. No T's whatsoever in RNA. This is one of the differences. Another difference is that it's single-stranded. So let's write that in. Single strand and no thymine, no T's. Okay, single strand and no thymine. Really important for us to know that and to know that difference. A codon is every 3 bases in a sequence of mRNA. So again, here's our codons. And an anticodon is 3 bases on tRNA. Here's mRNA, 3 nitrogen bases, UAG or CGA, is going to code for a codon. If we have 3 bases on a tRNA, that's going to be called an anticodon. And later we'll talk about how these two pair up, or base pair. But onto our main topic for today is transcription. And transcription is the first step to protein synthesis. We need to know, where does this happen? This occurs in the nucleus, and the purpose of it is to write the DNA or rewrite the DNA code as mRNA. Okay, very important. DNA cannot leave the nucleus. It's far too big, it's too important to go to the ribosomes where proteins are made. So, because DNA is too big to leave the nucleus, we need to get a smaller message out to the ribosome. The ribosome is where DNA is made. So in transcription, we're actually going to copy or rewrite a section of the DNA, one gene, as mRNA, and that mRNA can leave the nucleus and go to the protein, or go to the ribosome to make the protein. Our steps here to transcription, the first thing that happens is that enzymes will unzip a section of DNA. So here's my DNA in red and green. We open it up, and we'll copy one side of that gene, okay, to make a protein eventually. The second is the nucleotides use the side of DNA and base pair. So our DNA has opened up. Now we actually base pair mRNA with DNA to get this orange strand, and the orange strand is our new little messenger RNA, mRNA, or taking the message out of the nucleus. Our third step is that mRNA is complete. We're finished making the mRNA molecule. The mRNA will leave the nucleus through a pore in the nuclear membrane. Remember, we saw a picture or a video of the nucleus. We saw little holes inside of it. And the code is complete, but now we will need to translate. So our protein is not made yet. One thing I want you to add in is that it leaves the nucleus and goes to the ribosome. Leaves the nucleus and goes to the ribosome. You want to add that in. The other thing is, in your summary box, it says to transcribe the sequence into mRNA. They're giving you a strand of DNA. So if I have AUG for DNA, how would the mRNA pair up? Well, A's always pair with what? If this is DNA, now if I was making another strand of DNA, A would pair with T. But remember that when we're making mRNA, we have no T's. So mRNA, what we're now making, or base pairing with it, will actually have Actually, what I just realized is that that summary box, that question is wrong. We shouldn't have U's in DNA. So if you'll just actually skip that part, and we'll talk about that part